Martha,
Good afternoon, sir.
good evening. Sure. So we have finished off with cell division, and we are now left out on the most important parts of your NEET syllabus, the chapters of genetics. So, चार chapter है. We're going to do molecular basis of inheritance, that is about DNA, RNA, transcription, translation, principles of inheritance, that is about Mendel's laws of inheritance and chromosomal patterns, pedigree charts, etc. And then biotechnology and applications of biotechnology. तो आपके नीट में ये चार चैप्टर्स है. You can expect lots of marks coming from this part. Lot of MCQs will come from this part, tough part of the syllabus. So we are left out with these final four chapters. Let us start today with molecular basis of inheritance. Everything about the DNA we try to understand today. So let's talk about the DNA. So we begin with the DNA. It was in 1869, Frederick Miescher. He separated a cellular substance from the nucleus of pus cells and called it nuclein. तो न्यूक्लियस ऑफ पस सेल्स में से न्यूक्लिन नाम का केमिकल आइसोलेट किया था, and later on it was found nuclein has acidic properties and was called as nucleic acid. And then nucleic acid were found in two forms, DNA and RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. So DNA and RNA, two nucleic acids were discovered. What are the chemical composition of DNA and RNA? So DNA as well as RNA, of course you all have done it, consists of units which are called as nucleotides. So you have units which are called as nucleotides, and ये है nucleotides, a nitrogen base attached to it a sugar, and attached to it a phosphate. A nitrogen based sugar and a phosphate is called as a nucleotide. So the DNA is nucleotide, 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 and the other strand is also nucleotide, nucleotide, and both strands are nothing but polynucleotide strands. Both DNA strands are polynucleotide strands, and two DNA strands are polynucleotide strands. Now, what are the nucleotides made up of? The sugar is a pentose sugar called as deoxyribose sugar C5H10O4 or ribose sugar C5H10O5. अभी ये important है. This is the sugar. The sugar is a pentose sugar and a 5-carbon structure with a pentagon-shaped ring. So the sugar is a pentose sugar, and in the form of a pentagon-shaped ring, it has got first carbon, second carbon, third carbon, fourth carbon inside the ring, and the fifth carbon is outside the ring attached to the fourth carbon. And in the fifth position, there is an oxygen. So, how many carbons in the ring? 4 carbons inside the ring. Fifth carbon is attached to the fourth carbon, and it is a pentagon-shaped ring. It has got 5 carbons. So, it is pentose. It has got 10 hydrogens. So, it is C5H10 and O4. O4, there are 4 oxygens. If it is a deoxyribose sugar, which is DNA, then it is C5H10O4. But if it is RNA, then RNA is a ribose sugar, which is C5H10O5. So, what is the difference between deoxyribose sugar and ribose sugar? Instead of C5H10O4, it is C5H10O5. And what is the difference in the sugar actually? Here we have oxygen. So, rule of thumb, in the clockwise direction, you will have C1, and then C2, and then C3, C4, and C5 outside. So you have heard of it, oxy, para, meta, ortho. The first carbon which comes in the clockwise direction from the functional group. तो यहाँ functional group oxygen है, and the first carbon which comes in the clockwise direction is C1. At C1 is attached H and OH, and at C2 is attached H and H. H and H in case of a deoxyribose sugar. So, how many oxygens are there? 1, which is the functional group, OH at the second carbon, at the third carbon also OH, and attached to the fifth carbon is also an OH. So we have hydroxyl, hydroxyl, hydroxyl. So 3 oxygen हो गए, and functional group is the oxygen, which is the 4th oxygen. This is in case of deoxyribose sugar. In case of ribose sugar, at the second carbon, instead of H, there is an OH. So OH is present in case of the second carbon. तो second carbon पे hydroxyl group है, and that makes it oxygen, 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 and oxygen. 1, 2, 3, 4, 5 oxygens, and makes it ribose sugar C5H10O5. So ये नीट में बहुत commonly पूछते हैं, what is the difference between ribose sugar and deoxyribose sugar? Ribose sugar is C5H10O5. Deoxyribose sugar is C5H10O4 at the second carbon. In case of deoxyribose sugar is a hydrogen. In case of ribose sugar, there is a hydroxyl. तो second carbon पे hydrogen होगा, तो deoxyribose sugar, hydroxyl होगा, तो ribose sugar. So that is the difference of sugar. Phosphates are in the form of phosphoric acid, H3PO4. P double bond O, OH, OH, OH. These are phosphates. So, what is this entire structure here? H3PO4, C5H10O4. H3PO4, C5H10O4. And that is your alternately arranged sugars and phosphates. And then we have nitrogen bases in between. Nitrogen bases are cyclic compounds made of carbon, hydrogen, oxygen, and nitrogen. And ये है nitrogen bases. You have carbon, hydrogen, oxygen, and nitrogen. So they are organic, but they also have element nitrogen in them. Hence, these are called as nitrogen bases. So CHON, nitrogen की वजह से these are nitrogen bases. Of course, you have done it so many times. School में भी किया है. Double ring nitrogen bases and single ring nitrogen bases. Double ring nitrogen bases, purines, adenine and guanine. Single ring nitrogen bases are pyrimidines, thymine and cytosine. DNA has thymine. DNA has got thymine. RNA does not have thymine. It has uracil. So DNA has thymine or cytosine. RNA has uracil. This is your nitrogen bases. And a nitrogen base is attached to the first carbon. तो ये है नाइट्रोजन बेस, ये है आपका शुगर, ये है फर्स्ट कार्बन, और फर्स्ट कार्बन पे नाइट्रोजन बेस अटैच्ड होता है बाय अ बॉन्ड कॉल्ड एज अ ग्लाइकोसाइडिक बॉन्ड. सो ये है आपका शुगर एंड ऑक्सीजन C1, C2, C3, C4, C5, and attached to first carbon is the nitrogen base purine or pyrimidine by a bond called as a glycosidic bond. तो ये भी नीट में पूछते हैं, where is the nitrogen base attached to the first carbon of the sugar? What bond is that bond called as a glycosidic bond? And you have nitrogen base attached to it at the first carbon is the sugar, and ये है oxygen C1, C2, C3, C4, and at the fifth carbon is attached a phosphate. At fifth carbon is attached a phosphate, that is by a bond called as a phosphoester bond. So what is a bond called as a phosphoester bond? So nitrogen base is attached to first carbon and phosphate is attached to fifth carbon. Nitrogen base is attached by glycosidic bond. Phosphate is attached to fifth carbon by a phosphoester bond. And a nitrogen base plus a sugar is called as a nucleoside. Nucleoside. And a nucleoside, that is nitrogen base and sugar, attached to it phosphate at fifth carbon, is a nucleotide. So a nucleoside plus a phosphate group is called as a nucleotide. Nucleoside plus the phosphate group is called as a nucleotide. And sir, how is the base or linkage established? Sir, ये है आपका नाइट्रोजन बेस और ये है आपका नाइट्रोजन बेस, and sorry, ये आपका नाइट्रोजन बेस. Attached to the nitrogen base is the sugar. Attached to nitrogen base is the sugar at first carbon. And at the third carbon, there is a hydroxyl group. And at the fifth carbon, there is a hydroxyl group. And phosphate is in the form of H3PO4. So P double bond O, OH, OH and OH. तो होता क्या है, between the fifth carbon and the phosphate, hydrogen and hydroxyl condense to form a water molecule. Hydrogen and hydroxyl condense to form a water molecule. तो यहां पर भी hydrogen and hydroxyl condense to form a water molecule, and water molecule निकल जाता है. So now what are we left out with? We are left out with RCOR. Similarly, here also RCOR. What is R bond, C bond, O bond, R? R bond, C bond, O bond, R. RCOR, you have heard it in chemistry, is an ester. Is an ester. So phosphate is attached to fifth carbon by a ester bond, and that is a part of nucleotide. And phosphate is also attached to third carbon of the next sugar. Third carbon of the next sugar, again by RCOR bond, which is again an ester. So RCO, rather RCOR, ester bond, and RCOR, again an ester bond. So RCOR, ester bond, and similarly RCOR, ester bond, that is now called as a phosphodiester bond. What is that bond called as? A phosphodiester bond. So sugars and phosphates are attached alternately. ये है phosphate sugar, phosphate sugar, phosphate sugar, और sugar के fifth carbon के ऊपर होता है phosphate bond, which is a part of the nucleotide, and it is attached to third carbon of the next sugar also by ester bond. So phosphoester is called as a phosphodiester bond. So ऐसे phosphodiester bond कहते हैं. That is the way a chemical structure is attached. Third, a nitrogen base linked to a pentose sugar by a bond called N-glycosidic bond is called as a nucleoside. ये NCERT statements है. And you have adenosine or deoxyadenosine. Adenosine or deoxyadenosine are nucleosides. Guanosine or deoxyguanosine, depending on DNA or RNA. Then we have cytidine. So adenosine, deoxyadenosine, cytidine, or deoxycytidine. Deoxycytidine. And you have uridine, which is found in RNA, or deoxythymidine, which is found in DNA. DNA में thymine है. So what is the nucleoside called as? Deoxythymidine. RNA में deoxythymidine नहीं हो सकता है, या thymidine भी नहीं हो सकता है, because RNA does not have thymine. It has uracil. So what is a nucleoside called as? Uridine. That is called as a uridine. And in RNA, every nucleotide has an additional OH group present at second carbon in the ribose sugar. So what is the difference between DNA and RNA? RNA has got at the second carbon H, and DNA has got at the second carbon H. RNA has got at the second carbon OH, and that makes it C5H10O4 or C5H10O5. Also in RNA, uracil is found in presence of thymine, which is called as 5-methyl uracil. So सबसे पहले क्या आया था? RNA आया था नेचर में या DNA? What had evolved first, DNA or RNA?
RNA.
First had evolved RNAs, because सबसे पहले viruses थे, और viruses में RNA होता है. Many viruses have RNA. And viruses evolved to become bacteria and then other organisms, and RNA became DNA. तो सबसे पहले नेचर में कौन आया था? यूरासिल. And uracil is relatively unstable. Uracil unstable होता है, तो उसके फिफ्थ कार्बन पे एक मिथाइल ग्रुप आ गया, and that became 5-methyl uracil. And 5-methyl uracil is also called as thymine. 5-methyl uracil is also called as thymine, and thymine is a much more stable nitrogen base compared to a uracil. That is the reason DNA is a more stable molecule compared to RNA. RNA में तो OH group होता है second carbon पे. OH is reactive. It can form bonds. And OH की जगह H होता है DNA में. DNA is relatively more stable, and RNA has got uracil, which is unstable. DNA has got thymine, which is 5-methyl uracil, and is relatively more stable. Everybody understood that? Yeah. Just 1 second, guys. 1 second. Okay. So with that, we have analyzed the structure. Then we have the 3-dimensional structure called as the Watson and Crick model, which was based on X-ray diffraction technique produced by Maurice Wilkins and Rosalind Franklin. ये नीट में आया है. Who has given the X-ray diffraction technique? Maurice Wilkins and Rosalind Franklin. And what is the double helix model given by James Watson and Francis Crick, based on the X-ray diffraction technique produced by Maurice Wilkins and Rosalind Franklin? And what does the structure of the DNA look like? DNA is a long polymer of deoxyribonucleotides, and bacteriophages have, say, 4174 has 5386 nucleotides. Bacteriophage lambda has 48,502 nucleotide pairs. Escherichia coli has 4.6 into 10 raised to 6 base pair. ये नीट में पूछा गया है. How many base pairs are found in E. coli? 4.6 into 10 raised to 6, 4.6 million base pair. And haploid content of human DNA is 3.3 into 10 raised to 9 base pair. Haploid content. In other words, एक सेल के अंदर होते हैं दो डीएनए, एक मैटरनल डीएनए होता है और एक पैटर्नल डीएनए होता है, और एक पैटर्नल होता है. एंड एक हैप्लॉइड डीएनए में कितने बेस पेयर है? 3.3 into 10 raised to 9, 3.3 मिलियन बेस पेयर. सो मैटरनल में 3.3, पैटर्नल में 3.3, so a total of 6.6 billion base pair. 6.6 billion base pairs are there in one diploid cell. एक डिप्लॉयड सेल में 6.6 बिलियन बेस पेयर्स होते हैं. 6.6 बिलियन बेस पेयर्स. And this is your three-dimensional structure of the DNA. So DNA consists of two strands which are parallel to each other and are spirally twisted around the imaginary central axis, which is called as a DNA double helix. Neat point of view, what is our kind of DNA called? It is called as a BDNA. ये एनसीईआरटी में दिया नहीं है, but याद रखना, what is our kind of DNA called as a BDNA? It's a BDNA. There are 5 types of DNA: A, B, C, D, and Z. And ours is a type of DNA called as BDNA, which is a right-handed helix. Right-handed helix. And has 10 pairs of nucleotides inside a double helix. Double helix has got 10 pairs of nucleotides. So this is your Watson and Crick model. Now, what is the structure of each strand? According to Watson and Crick, DNA model consists of two long strands coiled around the imaginary central axis that is called double helix, which looks like a twisted ladder. And each strand consists of nucleotides, which we know already: deoxyribose sugar, phosphate, and nitrogen base. And sugar phosphates are the backbone, and the bases project inside. So who is the backbone of the DNA? Backbone of the DNA is sugar phosphate, sugar phosphate, sugar phosphate. And nitrogen bases are present inside. Nitrogen base अंदर होते हैं, connected by hydrogen bonds. And the successive nucleotides are connected by 3,5 phosphodiester bonds. Phosphate attached to 5th carbon of a nucleotide is joined to 3rd carbon of the next nucleotide. The nitrogen bases present on one strand pair with the nitrogen bases of the opposite strand. Adenine always pairs with thymine by 2 hydrogen bonds, and guanine always pairs with cytosine by 3 hydrogen bonds. This is called complementary base pairing. So always it is adenine pairing with thymine and guanine pairing with cytosine. AT के बीच में doublet, GC के बीच में triplet. AT doublets, GC triplet. This is called complementary base pairing. And then we have complement purine pyrimidine ratio. How much will be the purine pyrimidine ratio? Because base pairing is complementary. Base pairing is complementary. If you have a pyrimidine here, you automatically have a purine here. If you have a purine here, you automatically have a pyrimidine here. So if you count all the number of pyrimidines, thymine plus cytosine, 1, 2, 3, 4, 5, 6, 7. And if you count all the number of purines, 1, 2, 3, 4, 5, 6, 7. So 7:7. In other words, 1:1, which is called as a Chargaff's rule. What is Chargaff's rule? Chargaff's rule is that purine pyrimidine ratio is always 1:1. Chargaff's rule is A plus G upon T plus C is equal to 1. This is called as Chargaff's rule. which is true in case of DNA, but not necessarily true in case of RNA. RNA में Shargaf's rule constant नहीं है, but DNA में because of complementary base pairing, Shargaf's rule is always constant. Shargaf's rule is always constant. And एक चीज याद रखना, A plus T, or rather A plus T upon G plus C, that is adenine and thymine upon guanine plus cytosine, is always constant for a species. Constant for a species. Now, what is always equal to 1? A plus G, that is purine, upon T plus C, that is equal to 1, which we remember is called Shargaf's rule. A plus G upon T plus C. But yahan A pair with T and G pair with C, which is adenine thymine upon guanine cytosine, that ratio is not 1, but is always constant for a species. So A plus T upon G plus C is always constant for a species. ये याद रखना. And there are two ends. So sir, यहाँ check this here. Sir, ये है phosphate. Phosphate is attached by phosphodiester, 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 phosphodiester. So how many such phosphodiesters are there? A total of 3.3 billion phosphodiester bonds. If you remember, there are 3.3 billion nucleotide pairs. So 3.3 billion phosphodiester bond. And ये है आपका uppermost nucleotide. Uppermost nucleotide. यहाँ fifth carbon पे there is a phosphate. Fifth carbon पे phosphate. But इसके ऊपर कोई nucleotide नहीं है. ये तो last nucleotide है. इसके ऊपर कोई nucleotide नहीं है. So this phosphate is attached to fifth carbon of the nucleotide, but is not attached to third carbon. क्योंकि उसके ऊपर phosphate ही नहीं है. So this is phosphate attached only to fifth carbon. Everywhere else, phosphate is attached to third carbon, fifth carbon, third carbon, fifth carbon, third carbon, fifth carbon, third carbon, fifth carbon. So everywhere it is phosphodiester, 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 phosphodiester. But at one end, there is a free phosphate attached to only fifth carbon. So this is not phosphodiester, this is phosphomonoester. Everywhere else it is phosphodiester. Phosphodiester, diester, diester, diester. But this is phosphomonoester. So it is a free phosphate. And free phosphate is attached to which carbon? Fifth carbon. And that end is called as C5 end. C5 end पे free phosphate है, which is a phosphomonoester. And that is called as a C5 end. And everywhere phosphodiester, 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 phosphodiester. Now we come to the last nucleotide. Last nucleotide पे at the C1, C2, C3, at third carbon there is a hydroxyl group. Do we remember everywhere else at the third carbon the hydroxyl groups were utilized for the formation of water molecule to form phosphodiester bond. तो everywhere else यहाँ पर जो third carbon है उसका hydroxyl group was condensed with hydrogen to form a water molecule, and that is a RCOR, phosphodiester bond. So everywhere else hydroxyl is used up, but here at the third carbon, oxygen C1, C2, C3 and C4 and C5, at the third carbon there is a hydroxyl group which is not utilized, क्योंकि इसके नीचे कोई न्यू न्यूक्लियोटाइड है ही नहीं. न्यू न्यूक्लियोटाइड है ही नहीं, तो यहाँ नाइट्रोजन शुगर फॉस्फेट तो फॉस्फोडाइस्टर बॉन्ड बनता नहीं है. So there is no phosphate to combine because this is the last nucleotide, which means there is a free hydroxyl group. Everywhere else, hydroxyl groups were utilized to form water molecules, to form phosphodiester bond. But here there is a free hydroxyl group. इसका hydroxyl group का दूसरे hydroxyl group के साथ water molecule नहीं बनता है because there is no nucleotide here. So this is a free hydroxyl group. So free hydroxyl group is present at which carbon?
Third carbon.
At the third carbon. थर्ड कार्बन पे फ्री हाइड्रोक्सिल ग्रुप है, तो ये है ऑक्सीजन, ये है C1, ये है C2, ये है C3. At the third carbon there is a free hydroxyl group, and that end where you have free hydroxyl group is called the C3 end. So fifth carbon पे क्या था? Free phosphate, and that is called C5 end. And third carbon पे क्या है? Free hydroxyl, that is called C3 end. So we have third carbon पे free hydroxyl, C3 end, fifth carbon पे free phosphate, C5 end. And 3 end to 5 end is the polarity of the strand. Polarity of the strand is always from C3 end to C5 end. तो यहाँ C3 to C5 ऊपर जा रहा है या नीचे जा रहा है? ऊपर जा रहा है. So that strand is called as ascending strand. That strand is called ascending strand. Now check this here. ये strand के अंदर all the oxygens are facing upward. Oxygens are facing upward. So oxygen facing upward, this will become C1, C2, C3, C4, C5. And free phosphate is above because oxygens are facing upward. And yahan C1, C2 and C3 yahan, so hydroxyl is down and C3 end is downwards. But in the opposite strand, yahan oxygens are facing upward, but in the opposite strand, oxygens are facing downwards. Oxygen niche hai, and oxygen niche hai. So rule of thumb, the first carbon that comes in the clockwise direction is C1. So this becomes C2, this becomes C3, this becomes C4, and this becomes C5. So यहाँ third carbon पे क्या आ जाएगा? Free hydroxyl. So ये है oxygen C1, C2, C3 और third carbon पे free hydroxyl. So what is this end called as? C3 end. C3 end because there is a free hydroxyl. And yahan oxygen is facing downward. So ye hai C1, C2, C3, C4 and C5. And what is attached to C5? Free phosphate. Free phosphate. तो यहां पर कौन सा एंड आ जाएगा? C5 एंड. और यहां कौन सा एंड आ जाएगा? C3 एंड. Where is polarity always from? C3 to C5. Polarity is always from 3 to 5. So this strand is going ऊपर या नीचे?
नीचे.
Down. So this strand is rightly called as the descending strand. So in one strand, C3 is down, C5 is up. That is called ascending strand. In the other strand, C3 is up, C5 is down. So that strand is called as the descending strand. So both strands are parallel to each other, but in the opposite direction. They are parallel, but in the opposite direction. Hence, these are called as anti-parallel nature. Hence, they are called as anti-parallel. तो ये है आपके दोनों strands. ये है आपके दोनों strands. And एक strand में check this here. ये है C3. So this strand is going downwards. यहाँ arrow marks भी दिखाए हमने. This strand is going downwards. This strand is going downwards. from C3 to C5. तो ये कौन सा स्ट्रैंड है गोइंग डाउन?
descending strand.
Descending strand. And this strand is C3 going upwards.
ascending
strand.
ये कौन सा स्ट्रैंड हो गया? C3 to C5. Ascending strand. So one is a descending strand, one is an ascending strand. So both strands are parallel but in the opposite direction. So these are called anti-parallel in nature. ये दोनों strands anti-parallel हो गए. Then we have grooves. Grooves होते हैं. What is the grooves? Grooves, coiling of a double helix results in the formation of major or deep grooves. And twisting of strands around one another forms a minor or shallow groove, and which are arranged in an alternative manner. अब ये बहुत ध्यान से सुनना. Distance between two strands is called as a minor groove. Distance between two strands is called minor groove. तो ये जो groove है, what is this groove called? Minor groove. What is this groove called? Minor groove. Distance between two strands is called minor groove. So distance between two strands is called minor groove. And minor groove is the distance between two strands. ये ध्यान से सुनना. तो ये है डबल हेलिक्स, ये है माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव. So distance between two strands is minor groove. Minor groove is 12 angstrom units. Minor groove is 12 angstrom units. And distance in a double spiral. Distance in a double spiral. ये है एक स्पाइरल, उसमें ये है माइनर ग्रूव, ये है दूसरा स्पाइरल, ये है माइनर ग्रूव, और उनके बीच का जो डिस्टेंस है, that space is called as a major groove. तो एक डबल स्पाइरल का जो स्पेस है is called as a major groove. So major groove is 22 angstrom units. Major groove is 22 angstrom units. So what is distance between two strands called?
Minor groove.
Distance between two strands is called
minor groove.
Minor groove. Or minor groove is how much?
12 angstrom.
And ये भी है minor groove. Minor groove is 12 angstroms. ये है minor groove. Minor groove is 12 angstroms. और ये है distance in one double spiral. Distance between one double spiral. What is that distance called?
Major groove.
Major groove is how much?
22
angstrom.
22 angstroms. Now see this carefully. This distance is one spiral of one strand. One spiral of one strand. ये तो था. This was major groove. ये है major groove. ये distance है major groove, which is double spiral. Double spiral के बीच का distance जो है is called as major groove. Double spiral का जो distance है is called major groove. अभी मेरी उंगलियां ध्यान से देखना. See this carefully. What is this groove called?
Minor groove.
Minor groove. ये minor groove हो गया. और ये distance क्या हो गया?
Major groove.
Major groove. Major groove. Now see this carefully. See this carefully. Minor groove. And look at my fingers. minor groove 12 angstroms. This distance is major groove. Major groove 22 angstroms. But look at my finger. ऊपर वाले हाथ की लोअर फिंगर और लोअर वाले हाथ की अपर फिंगर, उसका जो डिस्टेंस है इज कॉल्ड मेजर ग्रूव, which is 22 angstrom. Now, if I remove one strand, मैंने lower strand ही निकाल दिया. So this is distance of one spiral of one strand. This is one spiral of one strand. One spiral of one strand. This is one spiral of two strands. One spiral of two strands, which is called major groove. Which is called major groove. One spiral of two strands is called major groove. Now, this is one spiral of only one strand. One spiral of one strand, which is minor groove plus major groove. Minor groove plus major groove. Now see it here in the diagram. What is this distance called?
Minor groove.
Minor groove.
Minor groove.
Minor groove. Minor groove. What is this distance called?
Major groove.
Major groove.
Now, minor is 12. Major is 22.
22.
Now, if I see this distance, check this. This and this. So, ये distance कौन है? One spiral of one strand. One spiral of one strand. एक strand का spiral है. एक strand का spiral. तो एक strand का spiral is a minor groove plus a major groove. One spiral of one strand. This is one spiral of one strand. One spiral of one strand. Or we are showing here, this is one spiral of one strand. One spiral of one strand. It is minor groove plus major groove. हाँ या ना? समझ में आया all of you? If you have understood, show a thumbs up. So this is one spiral of one strand, which is minor groove plus major groove. So how much was minor groove? 12 angstrom.
22.
How much is major groove?
22 angstrom.
So how much is this distance?
34 angstrom.
34 angstrom.
Plus 22, 34 angstroms. And 34 angstrom is called the pitch of the DNA. What is 34 angstrom called as? The pitch of the DNA. And one pitch of the DNA has in it 34 angstrom units, and देख इसको ध्यान से देखना, nucleotides. How many nucleotides are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, how many nucleotides? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 nucleotides are present. तो एक स्पाइरल में 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nucleotide है. So 10 nucleotides have how much distance? 10 nucleotides have how much distance?
34 angstrom.
34 angstrom.
34 angstrom.
34 angstroms.
34.
So tell me, one nucleotide has how much distance?
3.4
0.4
angstrom.
angstrom.
3.
Nucleotide has 3.4 angstroms. Also can be written as one nucleotide has 3.4 angstroms, also written as 0.34 nanometer. How much is one angstrom? 10 raised to minus 10. How much is a nanometer? 10 raised to minus 9. So 3.4 angstrom is same as 0.34 nanometer. So how much is a distance between two nucleotides? 0.34 nanometer. ये सब कुछ आपके एनसीईआरटी में दिया है, हाँ, ध्यान से याद रखना सब. Have you all understood this, all of you? And this is the pitch. तो ये pitch है. ये हो गया pitch. और एक pitch में minor groove plus major groove आ गया, which is 12 plus 22, which is 34, and has how many nucleotides in it? A total of 10. तो 10 nucleotides का distance कितना है? 34. तो एक nucleotide का distance कितना हो गया? 3.4 angstrom units or 0.34. Now tell me, what is the angle between two nucleotides?
360 upon 10, that is 36.
बराबर है. 360 से एक पिच बनता है ना. एक पिच एक सर्कल हो गया तो 360 डिग्री, उसमें 10 न्यूक्लियोटाइड है. So how much is the distance between two nucleotides? 36 degree. So distance between two nucleotides, rather angle between two consecutive nucleotides is 36 degree. And the diameter of the DNA is 20 angstroms. Diameter of the DNA is 20 angstroms. Have you understood? So neat point of view, minor groove is how many angstrom? 12. Major groove is how many? 22.
22.
How much is pitch?
34 angstrom.
34
angstrom.
angstrom.
How much is angle? One nucleotide?
36.
36.
One nucleotide का distance कितना है?
3.4.
3.4 angstrom or 0.34 nanometer. How much is angle between two nucleotides?
36
degree.
And how much is diameter of DNA?
20 angstrom.
20 angstrom.
Or 2 nanometer. ये सारे mathematical structures याद रखना. Are we clear with this, all of you? So this is the structure of the DNA, and then we'll just take a 2-minute break and go into a very beautiful logic called as the replication of the DNA, which is called semi-conservative method of DNA replication. ये बहुत important है. इस साल आपके नीट के पेपर में they had asked a question from replication. Of course, they asked prokaryotic. So we'll come to that. So we'll just take a 2-minute break and we'll come to that. Ciao. Yeah. So let's now get into the semi-conservative method of DNA replication. The process by which DNA molecule possesses exact copies or replica of itself is called DNA replication, which takes place inside the nucleus. And DNA कैसे replicate होता है? One mother DNA forms 2 daughter DNAs, in which one strand is an original strand. तो ये हो गया original strand. ये हो गया original strand. And this is the old strand. और ये strand बनता है which is a new strand. So dotted वाला कौन सा है? Marked वाला कौन सा है? New strand. And which is this strand. This strand is the old strand. तो ये वाला old strand है, ये वाला old strand है, और ये वाला जो बनता है, this one is formed is the new strand. This strand formed is the new strand. So what is this mechanism called as? It is called semi-conservative method of DNA replication. Semi-conservative method of DNA replication. Y'all tell me, कौन से phase of cell division में DNA replicate होता है?
S phase.
Phase में.
S phase of interphase. S phase of interphase, DNA will replicate in the S phase of interphase. So सबसे पहले, how will replication begin? There is origin or initiation points. DNA replication begins at specific sites which are called as origin points or initiation points. And there are several origin points. At the origin point, there is DNA molecule is split by activity of enzyme, incision enzyme, which is also called endonuclease. And endonuclease will create an incision or nick. तो ये है आपका DNA और ये है DNA, उसके बीच में एक incision आता है. And incision is made by incision enzyme, also called endonuclease. So endonuclease will cut which bonds?
Phosphodiester.
Phosphodiester.
ये कौन सा bond था? Phosphodiester bond. If you remember, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. So incision enzyme will break which bond?
Phosphodiester.
Another enzyme called as helicase. कौन सा enzyme? Helicase. Helicase will come and start breaking the hydrogen bonds. Helicase will break the hydrogen bonds. So, who breaks phosphodiester bond?
Endonuclease.
Who will break hydrogen bonds?
Helicase.
So which are the two enzymes? Incision enzyme, which breaks phosphodiester bond, is endonuclease, and DNA unwinding protein, which breaks hydrogen bonds, is helicase enzyme. And helicase is also called DNA unwinding protein, also called replication protein. And because of which the two DNA strands which were coiled will now uncoil and will look like this. Will look like an inverted Y. Will look like an inverted Y. So जैसे helicase breaks hydrogen bonds, the two strands uncoil and looks like an inverted Y, which is called as the replication fork. What is the fork called as? Replication fork. और उसमें दोनों स्ट्रैंड सेपरेट हो गए हैं, दे आर हेल्ड टुगेदर. अभी व्हाट इज द प्रॉब्लम? दोनों स्ट्रैंड वापस नजदीक नजदीक आ जाएंगे तो उनके बीच में वापस हाइड्रोजन बॉन्ड बन जाएंगे. So the two strands which have separated by helicase are to be held in their position. तो उनको पकड़ के रखना होगा. And they are held in their positions by an enzyme called as SSBP, single strand binding protein. Single strand binding protein, SSBP, single strand DNA binding protein or helix destabilizing protein, will prevent the separated strands from coiling again. तो separated strands को अलग रखने के लिए कौन सा एंजाइम है? SSBP, single strand DNA binding protein. Single strand DNA binding protein. So once again, who breaks phosphodiester bond?
Endonuclease.
Endonuclease. Who breaks hydrogen bonds?
Helicase.
Helicase.
Who does unbinding? Helicase.
Helicase only.
Binding is done by helicase. Who holds the two strands in their position?
SSBP.
SSBP.
SSBP, single strand DNA binding protein. And now, अभी each separated strand is called as a template or mold. तो ये जो separated strand है ये mold और template है और उसके ऊपर बनेगा new strand. उसके ऊपर बनेगा new strand. So this will become old strand, new strand, old strand, new strand. So what will this become? Old strand. And what will this become?
New strand.
New strand. तो ये हो जाएगा semi conservative method of replication. And what is this old strand also called as template? अभी template के ऊपर बनता है एक छोटा सा molecule called as RNA primer molecule. An RNA primer molecule is a small RNA made with the help of an enzyme called RNA primase enzyme. तो RNA primase enzyme से उसके ऊपर बनता है a molecule called RNA primer molecule. And then RNA primer molecule attracts complementary nucleotides from the nucleoplasm. And this is done by DNA polymerase enzyme. So, nucleoplasm में नए नए न्यूक्लियोटाइड्स आके अटैच होते हैं आरएनए प्राइमर मॉलिक्यूल को. तो ये है टेम्पलेट. दिस इज टेम्पलेट और डीएनए, ओल्ड डीएनए. once again, this is template और ओल्ड डीएनए. उसके ऊपर आ गया आरएनए प्राइमर मॉलिक्यूल. और उसके ऊपर आ गया न्यू स्ट्रैंड. Who will form RNA primer molecule? Which
RNA primase.
RNA primase.
RNA primase. And who forms new strand?
DNA polymerase.
DNA polymerase.
New strand is made with the help of DNA polymerase. New strand is made with the help of DNA polymerase. तो ये है टेम्पलेट. टेम्पलेट बीच में आ गया आरएनए प्राइमर मॉलिक्यूल. एंड उसके ऊपर आ गया न्यू स्ट्रैंड. न्यू स्ट्रैंड इज मेड बाय डीएनए पॉलीमरेज. न्यू स्ट्रैंड इज मेड बाय डीएनए पॉलीमरेज. नाउ देयर इज वन प्रॉब्लम. DNA polymerase will construct new strand always from C5 end to C3 end. DNA polymerase makes new strand from C5 end to C3 end. So, what will be the direction of template?
C3 to C5.
C3 to C5.
C3 to C5. क्योंकि जब दोनों strands बनेंगे, they have to be complementary to each other and anti-parallel to each other. So, DNA polymerase always goes from C5 end to C3 end of new strand. Means it will go from C3 end to C5 end of template. तो एक strand पे possible है 3 to 5 of template means 5 to 3 of new strand. But दूसरे strand पे polarity क्या थी? ये template पे है polarity 5 to 3. So can you have DNA polymerase going this direction 3 to 5?
No.
It cannot go from 3 to 5. It always goes from 5 to 3. तो एक strand पे DNA polymerase move होगा. तो दूसरा स्ट्रैंड जिसका पोलैरिटी 5 to 3 है, इसके ऊपर डीएनए पॉलीमरेज 3 to 5 तो नहीं जा सकता है. इट कैन ओनली गो फ्रॉम 5 to 3. तो हाउ विल इट सप्लाई न्यूक्लियोटाइड्स? So it will supply some nucleotides to one strand. तो 3 to 5 polarity और यहाँ 5 to 3 polarity में कुछ nucleotides arrange हो गए, कुछ nucleotides. After which, from here, some DNA polymerase takes a U-turn and supplies nucleotides even to the other strand, but in the opposite direction. तो ये कौन सा डायरेक्शन में डीएनए पॉलीमरेज जा रहा है? ऊपर. और यहां से डीएनए पॉलीमरेज कौन से डायरेक्शन में जा रहा है? नीचे. नीचे जा रहा है. तो फ्रॉम विच पोलैरिटी इट इज गोइंग? 3 to 5 of template means 5 to 3 of new. So golden words, DNA polymerase always goes from 5 to 3 of new strand, means 3 to 5 of template. Other words, other side, it goes in the opposite direction from 3 to 5 of template, means 5 to 3 of new strand. So golden words, नया स्ट्रैंड का पोलैरिटी हमेशा कौन सा होता है?
5 to 3.
5 to 3. और टेम्पलेट का पोलैरिटी अपवर्ड होगा या डाउनवर्ड होगा? पोलैरिटी कौन सा होगा?
3 to 5.
3 to 5. So template polarity is always 3 to 5. Template polarity is always 3 to 5. And new strand polarity is always 5 to 3. New strand polarity is always 5 to 3. Clear? All of you? Now, what will happen? Some DNA polymerase goes upwards from 5 end to 3 end. और यहां कुछ नहीं बना. फिर यहां से U-turn हुआ. And new strand was constructed downwards. But while new strand is constructed downward, remaining DNA polymerase continues to move upward. तो यहां समझो कुछ न्यूक्लियोटाइड्स आए, यहां कुछ भी न्यूक्लियोटाइड्स नहीं आए थे. फिर ये न्यूक्लियोटाइड और ये न्यूक्लियोटाइड साइमल्टेनियसली बने. तो this strand is formed earlier and this strand is formed later. Once again, some nucleotides, no nucleotides. Now, some nucleotides, new nucleotides. Some nucleotides, new nucleotides. तो ये पहले बना, फिर ये सेकंड बना, फिर ये थर्ड बना. तो have you understood? In one strand, replication is earlier. In the other strand, replication is later. In one strand, replication is earlier. In the other strand, replication is later. तो क्या क्या पहले बना? पहले ये बना, यहां कुछ नहीं बना, फिर ये बना, तो साथ में ये बन गया, फिर ये बना, तो साथ में ये बन गया. तो एक स्ट्रैंड में रेप्लिकेशन पहले होता है, दूसरे स्ट्रैंड में रेप्लिकेशन बाद में होता है. सो वन स्ट्रैंड इज लीडिंग, one strand is leading, other strand is lagging. So यहाँ one gene बन गया, यहाँ no genes बना. अब यहां 1 जीन बना तो यहां 2 जीन बन गए. अब यहां 2 जीन बना तो यहां 3 जीन बन गए. So 1 gene, no gene. 2 genes, 1 gene. 3 genes, 2 genes. So in one strand replication is early and the other it is not there. Now this 1 gene, no gene. First gene, second gene. Second gene. Second gene, third gene. तो साइमल्टेनियसली क्या क्या हुआ? फर्स्ट जीन, नो जीन. फर्स्ट जीन, सेकंड जीन. सेकंड जीन, थर्ड जीन. तो साइमल्टेनियसली फर्स्ट जीन, नो जीन. अब यहां का फर्स्ट जीन और यहां का सेकंड जीन साइमल्टेनियसली हुआ. साइमल्टेनियसली सेकंड जीन हियर एंड फर्स्ट जीन हियर. Now simultaneously third gene here and second gene here. So this strand replication is earlier, this strand replication is later. This strand replication is earlier, this strand replication is later. So one strand is always called as leading strand. One strand is leading strand. And other strand जिसमें replication बाद में होता है is called as lagging strand. So one strand is called leading strand. Leading strand is formed in which template?
3 to 5.
3,
3
5.
to 5 template में leading strand बनता है upward direction. And 5 to 3 template में lagging strand बनता है, वो भी downward direction. So 3 to 5 template in the upward direction was formed the leading strand, and 3 to 5 but in the downward direction is formed the lagging strand. And leading strand is continuous. Leading strand continuous बना. 1 gene, 2 gene, 3 gene, so it was formed continuously. But lagging कैसे बना? First gene, फिर बना second gene, फिर बना third gene. तो leading बना continuous और lagging बना, ये बन गया continuous और ये lagging बना first gene, second gene, third gene. So have you understood? Leading strand is formed continuously, but lagging strand is formed first gene, second gene, third gene. तो lagging strand छोटे छोटे fragments में बनेगा. Leading continuous बनेगा, lagging छोटे छोटे fragments में बनेगा. And वो जो छोटे छोटे fragments होते हैं lagging के are called as Okazaki fragments. What are the fragments called? Okazaki fragments. So leading strand is continuous from 5 to 3 polarity along template having 3 to 5 polarity, and lagging strand is formed from 5 to 3 polarity wala template but in the opposite direction in the form of fragments which are called as Okazaki fragments, and this will result in template, leading strand, continuous, lagging strand in the form of fragments which are called as Okazaki fragments. तो ये fragments कौन से fragments है? Okazaki fragments. And end of replication ये बन जाएगा, ये बन गए template, ये बन गया continuous strand और ये strand बन गया fragments called Okazaki fragments. अभी you cannot have a fragmented DNA. So Okazaki fragments are stuck to each other. Okazaki fragment को चिपकाया जाता है with the help of an enzyme called as DNA ligase enzyme. Which enzyme? DNA ligase enzyme. DNA ligase enzyme will stick all the Okazaki fragments and make the other strand also become continuous. अब ये तो नया स्ट्रैंड बन गया. ये बीच में कौन सा मॉलिक्यूल था?
RNA primer.
RNA primer molecule. याद है ना? RNA primer made with the help of primase. Now, once the new strand is formed, an enzyme
RNA primer.
called RNase will come and remove the RNA primer molecule. RNase will come and remove the RNA primer molecule. तो RNA primer molecule को निकाल दिया जाता है with the help of RNase. That means now you have only two strands and दोनों strands complementary है. दोनों स्ट्रैंड कॉम्प्लीमेंट्री है, तो इनके बीच में क्या बन जाएंगे? इनके बीच में बन जाएंगे हाइड्रोजन बॉन्ड्स. And because of hydrogen bond formation, the two strands will start coiling around each other. The two strands will start coiling around each other because of hydrogen bond formation. And what are these two DNAs called as? Two daughter DNAs. So these are two daughter DNAs. So what is the nucleotide sequence? ये था CG. अब old C को मिल गया new G और old G को मिल गया new C. So this is old C, this is new G. This is old G, this is new C. So eventually CG has become CG, has become CG. Similarly, TA, old T gets new A, old A gets new T. So TA becomes TA, which will become eventually TA. Similarly, AT will become AT, will become AT. So what is the sequence of old strand? CG, TA, AT, GC, TA, CG, AT, TA. And what are the sequences of new strand? CG, CG, TA, TA, AT, GC, GC, TA, CG, AT, TA. So both daughter DNAs are genetically same and they are identical to the mother DNA. So what is this phenomenon called as? Semi-conservative method of DNA replication. This is called semi-conservative method of DNA replication. Clearly all of you understood this. And then we have packaging of the DNA. Packaging of DNA. What is the length of the DNA double helix? Length of the DNA double helix is 2.2 meters. अभी ये कैसे मिला आपको? What are the distance between two consecutive base pair?
0.34 nanometer.
0.34.
याद है 3.4 angstrom unit और 0.34 nanometer. 0.34 nanometer अभी किया था हमने. So multiply 0.34 nanometer with the total number of base pair. So total number of base pair हमने किया था. How many is total number of base pair?
6.6 into 10 power 9.
6.6 into 10 raised to 9 base pair. So 6.6 into 10 raised to 9 multiplied by 0.34 nanometer, which is 0.34 into 10 raised to minus 9. So 6.6 into 10 raised to 9 into 0.34 into 10 raised to minus 9. So 10 raised to minus 9 and 10 raised to 9 will cancel each other. And 6.6 into 0.34 comes out to be approximately 2.2 meters. Have you understood this, all of you? So 6.6 into 10 raised to 9 multiplied by 0.34 into 10 raised to minus 9, 10 raised to minus 9, 10 raised to 9 gone, 6.6 into 0.34 turns out to be 2.2 meters. That is the length of the DNA. And this DNA has to be accommodated in a nucleus which is 10 raised to minus 6 meters. तो 1 माइक्रोन, एक न्यूक्लियस 1 माइक्रोन का होता है, उसमें 2.2 मीटर लंबा डीएनए डालना है. So such a long DNA has to be accommodated in a very small nucleus. For that, it is condensed and coiled and supercoiled to fit into the nucleus. So how does this condensation take place? In eukaryotes, packaging or organization of DNA is very complex. And for that, there are histone proteins. Histone proteins are proteins which are made of amino acids. तो ये amino acids का प्रोटीन होता है, connected by peptide bonds. और उसमें ज्यादा करके अमाइनो एसिड होता है. और अमाइनो एसिड्स कितने टाइप के होते हैं नेचर में? 20 टाइप्स. बट उसमें ज्यादा करके अमाइनो एसिड होता है लाइसीन एंड आर्जिनिन. लाइसीन एंड आर्जिनिन. And lysine and arginine are basic amino acids. Arginine and lysine are basic amino acid which carry positive charge in their side chain. If they are basic amino acids, they have a positive side in their side chain, and DNA is negatively charged. DNA is negatively charged. So negatively charged DNA will coil around positively charged histone proteins. Opposite charges attract each other. So negatively charged DNA will coil around positively charged histone proteins, and that is the way DNA will coil around the histone proteins. So histone proteins के बाद positive charge है, DNA जो negative charge है will coil around it, and it will form उसमें 8 histones, 2 each of H2A, H2B, H3 and H4 get organized to form a histone octamer core. So this is an octamer core. 2 H2A, 2 H2B, 2 H3 and 2 H4. They form one cube, and this is called octamer core. उसके ऊपर होता है 1 turn and 3/4 turn. 1/3/4 turn of DNA. So 1/3/4 turn of DNA is there around a histone protein. 1/3/4 turn of DNA is there around a nucleosome core of histone proteins, and it has in it adenine pairs with thymine, thymine pair with adenine, guanine pair with cytosine, cytosine pair with guanine. ऐसे करके nucleotide pairs है. How many nucleotide pairs? In 1/3/4 turn, you have a total of 146 base pair. 146 base pair. So tell me one thing, यहाँ 1 turn और 3/4 turn. 1/3/4 turn में कितने base pairs है? 146 base pairs. और DNA को इसके ऊपर चिपकाने के लिए होता है a protein called as H1 protein. So totally कितने proteins हो गए? Octamer of 8 and H1 protein. So a total of 9 histone proteins. 9 histone proteins. So what is the octamer of 8 called as? Nucleosome core. उसके ऊपर 1/3/4 turn of DNA 1/3 ton of DNA with 146 base pairs. उसके ऊपर H1 to stick it, and that is called as one nucleosome core, and ये है one nucleosome core. Nucleosome core, nucleosome core, nucleosome core, nucleosome core, nucleosome core, nucleosome core. इसके ऊपर कितना टन ऑफ डीएनए है?
1 3/4 ton.
1/3 टन. ये कितना डीएनए है? 1/3 टन. ये कितना डीएनए है? 1/3 टन. ये कितना डीएनए है? 1/3 ton. Or what is that 1/3 ton of DNA called? It's called core DNA. What is that DNA called? Core DNA. Core DNA में बेस पेयर कितने हैं?
146.
146.
और दोनों न्यूक्लियोसोम कोर डीएनए के बीच में होता है डीएनए विच इज कॉल्ड लिंकर डीएनए. उनके बीच में जो डीएनए है इज कॉल्ड एज लिंकर डीएनए. And linker DNA has in it 54 base pairs. So, ये है कोर डीएनए और ये है लिंकर डीएनए. कोर डीएनए में कितने बेस पेयर है?
146.
और लिंकर डीएनए में कितने बेस पेयर है?
54.
तो टोटल कितने हो गए?
200.
200 बेस पेयर्स. 200 बेस पेयर. तो एक कोर डीएनए 146 बेस पेयर और एक लिंकर डीएनए 54 बेस पेयर which is 200 base pair is called as one nucleosome. ये हो गया एक न्यूक्लियोसोम. तो सिमिलरली ये हो गया दूसरा न्यूक्लियोसोम, ये हो गया तीसरा न्यूक्लियोसोम, ये हो गया चौथा न्यूक्लियोसोम, ये हो गया पांचवा न्यूक्लियोसोम. न्यूक्लियोसोम, न्यूक्लियोसोम, न्यूक्लियोसोम. How many base pairs in one nucleosome?
200.
200. How many here? 200, 200, 200, 200, 200, 200, 200 base pair. ऐसा this structure is bead string, bead string, bead string, bead string, bead string, bead string. ये अभी किया था सेल डिवीजन में. This is called as one chromatin thread. One chromatin thread. तो एक सेल में न्यूक्लियस, उसमें बीड स्ट्रिंग, बीड स्ट्रिंग, बीड स्ट्रिंग, बीड स्ट्रिंग. ये हो गया पैटर्नल डीएनए. सिमिलरली बीड स्ट्रिंग, बीड स्ट्रिंग, बीड स्ट्रिंग, बीड स्ट्रिंग, बीड स्ट्रिंग, बीड स्ट्रिंग. ये हो गया मैटर्नल डीएनए. तो कैन यू ऑल टेल मी टोटली हाउ मेनी बीड्स विल बी देयर?
Sir, 6.6 into 10 raised to 9 upon 200.
6.6 into 10 raised to 9 base pair. Divide by 200. If you remember, one bead in string is 200. So that is 6.6 upon 2 into 10 raised to 7, which is 3.3 into 10 raised to 7 beads are there. Can you imagine कितने beads है? 3.3 into 10 raised to 7 beads are there, which is 88 histone proteins. And all this 3.3 into 10 raised to 7 beads are packed into a nucleus which has a diameter of 10 raised to minus 6 meter, 1 micron. तो ये कितने माइक्रोस्कोपिक लेवल के ऊपर बीड्स है, कैन यू इमेजिन? इतना साइंटिस्ट ने एनालाइज किया है. बाय द वे, दिस इज बीड स्ट्रिंग. नाउ you will understand better what will happen during cell division. Bead string, bead string, bead string, bead string, bead string, maternal, bead string, bead string, bead string, bead string, bead string, paternal will replicate. और याद है, एस फेज में डीएनए भी रेप्लिकेट होता है, हिस्टोन प्रोटीन्स भी रेप्लिकेट होते हैं. So there will be now 2 bead string, bead string, bead string, bead string, bead string maternal, and 2 bead string, bead string, bead string, bead string paternal. And this will become 2N4C. याद आया ये अभी? So this is found in the S phase. So imagine 3.3 into 10 raised to 7 beads will become 6.6 into 10 raised to 7 beads. All found in the nucleus. And then what will bead string, bead string, bead string, bead string become? Bead, 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 bead. String, 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 string. Which if you remember is called as nucleosome fiber. And that will coil and supercoil to become, if you remember, solenoid, like a telephone wire, solenoid. And solenoid will coil to become super solenoid. And super solenoids will form chromatids. तो ये आ गया bead string, 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 bead string. और यहाँ आ गया bead string, bead string, bead string, bead string, bead string. By the way, ये दोनों कौन से DNAs है? These are daughter DNAs. और हमने अभी किया, daughter DNAs are genetically the same. And they are identical to mother. और उनके बीच में आ जाएगा सेंट्रोमियर और बाजू में आ जाएगा काइनेटोकोर. And this is formed in prophase. Metaphase there or on the equator. Anaphase में centromere will break. And daughter DNAs go to opposite sides in telophase. Then it will form 2 cells. What will both cells have in them? Daughter DNAs. And daughter DNAs are genetically the same. So one zygote has become 2 blastomeres which are genetically the same. And if you remember, if 2 blastomeres separate, they will form identical twins. Why? Because we just did it. DNA replication में क्या हुआ? One mother DNA became 2 daughter DNAs which are genetically the same. Have you understood this? So this is the beautiful packaging of the DNA. सबको समझ में आया? All of you? चलो, great. So next session we go into the most important part of the chapter, RNA and protein synthesis, transcription and translation. वो वाला पार्ट बराबर से पढ़ के रखना NCERT से और नोट से. I'm going to send you the notes for this chapter also and today's lecture also. See it carefully and prepare from the notes and the NCERT in the next lecture. Okay? Chalo. Bye-bye all of you. Take care, beta.
Bye.
Bye, sir. Thank you.
Sir,
Bye,
to
sir.
the division also you haven't sent the notes.
Sorry, beta.
Sir, cell division also you haven't sent the notes. You're just in the
I'm sending you the notes for both. I'll send